Good morning, students. We are starting our industrial microbiology syllabus further. In this video, we will discuss fermentation media. Fermentation media means the media that are used for fermentation. In fermentation media, we provide the nutrients for growth of microorganisms as well as for product formation. Different components are used in this fermentation media as nutrients for the fermentation microorganism and the substrate for product formation. In this topic, we are going to discuss which type of nutrients or elemental requirements are provided into the fermentation media. If we want the fermentation to be commercially successful, then several factors must be considered. A commercially successful fermentation requires several factors, including efficient microbial strain. That means a good production strain is very much important. Suitable raw materials or best possible medium for the fermentation, maintaining optimum environmental conditions during the fermentation, and monitoring and controlling the parameters. If we maintain all this, then we get good amount of product accumulation. But this accumulated product also should be isolated and purified. For that, we need good recovery procedure. So for commercially successful fermentation, we require suitable raw materials or the best possible medium. So fermentation medium is highly important for this. Nutrients that are required for fermentation media depend on various factors. The type of fermentation organisms means whether the organism is heterotrophic or it is autotrophic, or does it require some complex growth nutrients, or is it an auxotroph. All these facts should be taken into consideration. So the type of fermentation organisms are important. The type of fermentation process to be used is also important, because we have discussed different types of fermentations. In that, two major types of fermentation: batch fermentation and continuous fermentation. Designing the medium for batch fermentation is different than designing the medium for continuous fermentation. So the type of fermentation process also must be considered while designing a fermentation medium. Types of nutrients present in the fermentation media always determine the yield of the product. Which type of nutrients are given in the medium, that will determine how much amount of yield will be obtained. There are two uses of fermentation media. Actually, there are two types of fermentation media, as inoculum media and fermentation media. Inoculum media are also called as growth media. Growth media or inoculum media, and the second type is fermentation media. Growth media or inoculum media contain low amount of nutrients. Why the amount of nutrients is low in inoculum media? This is because in inoculum media we desire only cell growth. Therefore, we don't desire any product accumulation or product formation in inoculum media. Therefore, inoculum media contain only the amount of nutrients necessary for cell division to increase the cell number. Fermentation media contain high amount of nutrients. This is because in fermentation media we are aiming at product accumulation. Product is produced from the substrate and the substrate is provided through fermentation media. So if you provide a large quantity of substrate, then there are chances of getting large quantity of the product. For this purpose, we add excess of nutrients into the fermentation medium, and therefore fermentation media contain high amount of nutrients. During fermentation of alcohol, yeast requires 12 to 13% carbon in the medium to give maximum amount of alcohol, while growth of yeast requires 1% carbon. Suppose you just want the growth of yeast cells, then you can add only 1% carbon source in the medium, because only growth is required. The yeast cell should multiply. That is our purpose. But when you want the yeast cell to produce alcohol, then you have to increase the carbon source. You have to increase the amount of carbon source to 12 to 13%, so that you will get maximum production of alcohol. Nutrients that are required for fermentation media depend on the type of fermentation organism, the type of fermentation process, and the type of nutrients that are present in the fermentation media. This is a repetition, but it was important here. Role of fermentation media. When these microorganisms are incubated at the desired optimum conditions, they enjoy luxurious metabolism. It creates metabolic stress on the organisms. When the organisms are growing in a medium that has ample of nutrients, that has high quantity of nutrients, they become greedy and they enjoy luxurious metabolism. And due to high rate of metabolism, there is metabolic stress on the organisms. The amount of substrate given to microbes should not reach inhibitory concentration levels. The amount of substrate given to the microbes should not reach inhibitory concentration levels, because excess substrate inhibits vital enzymes and may result in death of cells. This should be kept in mind, that if you go on increasing the amount of substrate, because substrate is converted to product and we want high quantity of product, so add high quantity of substrate. This is the policy, but it is not always true, because beyond certain limit, if you add excess of substrate or if you provide excess of substrate, then it becomes inhibitory and it inhibits the enzymes, and therefore when the enzymes are inhibited, it leads to death of cells. Excess substrate may increase osmotic pressure and affect enzyme activities in the cell. Suppose in alcohol fermentation, sugar is converted into alcohol. So we add 12 to 13% of sugar to get maximum quantity of alcohol. Then why limit to 12 to 13%? Why we should not add 15%, 20%, 25% sugar? What will happen then? If you add excess of sugar, it will increase the osmotic pressure, and this increased osmotic pressure will affect the enzyme activities in a cell and it will lead to death of cells. So this thing should be kept in mind. Amount of substrate added should be enough, but not very excess. The amount of substrate added should be such that it will be tolerated by the microorganism, and then the microorganisms will act on the substrate and will convert it into the product. Now we will discuss the types of fermentation media. There are two types of fermentation media, as synthetic media and crude media. Synthetic media are useful in the field of research. In synthetic media, each and every component is chemically defined and known. The exact composition of nutrients is predetermined. To construct a synthetic media, you have to use standard chemicals. Suppose you want to add carbon source, then you should use purified sugars, standard purified sugars. If you want to add nitrogen source, then you have to use standard inorganic salts, of which the purity is known. Exact quantity of each component in the medium, that is also known, and such media are called as synthetic media. The second type of fermentation media are crude media. Crude media contain crude components, and as the components are crude, the exact chemical composition is not known. For example, a media is designed with molasses as a component. Exact chemical composition of molasses is not known. So a medium containing molasses will be a crude medium. A medium containing peptone will also be a crude medium. A medium containing sulfite waste liquor will be a crude medium. So if you add crude components of which exact purity or exact chemical composition is not known, such a medium will be called as crude medium. Crude media are commonly used for fermentation because they are economical and they give higher yields. Synthetic media are not commonly used because synthetic media are costly and the yield is also not very high. The fermentation media provide nutrients and energy for growth of microorganisms. They should provide nutrients for two things: growth as well as for energy, and the substrate for product synthesis in a fermenter. Means the fermentation media provide nutrients for growth, they provide nutrients for energy, and they also provide nutrients for product synthesis. Fermentation media consists of major and minor components. Major components include carbon source and nitrogen source. Minor components include inorganic salts, vitamins, growth factors, antifoams, buffers, DO, that is dissolved oxygen, and other dissolved gases, growth inhibitors and enzymes. Major components are so named because their quantity is large, and minor components are added in a smaller quantity in the medium. Now we will discuss the components of fermentation media one by one. The first component that we will discuss is water. Water is a major component of fermentation media because fermentation media, most of them fermentation media are liquid media. Fermentation media contains 70 to 90% water. How much amount of water is there in the medium? 70 to 90% water. Water is also required for cleaning and sterilization. It supplies trace elements and dissolved salts. The pH and dissolved oxygen of water is important. Whenever you select water or whenever you choose water source for the fermentation medium, then you should check the water for its pH, for its dissolved oxygen content, for its color, odor and mineral composition. These 5 things are very critical for selecting the water source for the fermentation medium: pH, DO, color, odor and mineral composition. Color, odor and mineral composition is important for brewing. Means suppose the product is beer or even if the product is wine, then color, odor and mineral composition is highly important. The availability of good cool water of constant composition dictates the location of fermentation industry because required water quantity is very high, and therefore good cool water of constant composition dictates the location of fermentation industry. The second important component is the component added as an energy source. Most fermentation organisms are chemoheterotrophic. This is the classification based on nutrition. So most fermentation organisms are chemoheterotrophic. They derive energy by oxidation of chemicals. For most organisms, the source of carbon and the source of energy is the same. So whatever carbon source you add to the fermentation medium, it also acts as a source of energy for most of the organisms. Now, the carbon source. The selection of carbon source depends on the availability as well as cost of raw material. Which two things are important? Carbon source is required in large quantity. So the cost of carbon source affects the total cost of fermentation medium, and therefore one should choose the carbon source based on the availability as well as the cost of raw material. In most of the fermentation media, crude source of carbon is added. Which different carbon sources are there? Simple carbohydrates and complex carbohydrates. Simple carbohydrates like sugars, semi-purified polysaccharides or sugar alcohols are added. Sources of simple carbohydrates are blackstrap molasses, corn molasses, wheat molasses, sulfite waste liquor, hydrol, corn sugar molasses, cannery waste, etc. Source of complex carbohydrates are starch, corn, rice, rye, millet, wheat, potatoes, etc. Source of cellulose are corn cobs, straws, wood waste, sawmill dust, etc. So, carbon source is selected based on the availability. It should be available locally as well as the cost of raw material. Carbon sources are generally crude in nature. Carbon sources can be further divided into two types as simple carbohydrates and complex carbohydrates. Simple carbohydrates, for example, simple sugars, semi-purified polysaccharides or sugar alcohols. For example, different types of molasses: blackstrap molasses, corn molasses, beet molasses, sulfite waste liquor, cannery waste, etc. Complex carbohydrate sources include starch, corn, rice, rye, millet, wheat, potatoes. Sometimes carbohydrate is added in the form of cellulose. So cellulosic wastes like corn cobs, straws, wood waste, sawmill dust, etc. can be used as a carbon source in the fermentation medium. Then the next nutrient component is nitrogen source. Most industrial microorganisms utilize organic as well as inorganic nitrogen sources. Inorganic nitrogen sources are salts of urea, ammonia, ammonium salts and nitrates. Organic nitrogen sources include amino acids, proteins, etc. Nitrogen source is of two types: organic nitrogen source and inorganic nitrogen source. Organic nitrogen sources are amino acids and proteins. Inorganic nitrogen sources include various inorganic salts like ammonia, ammonium salts and nitrates. When fermentation organisms are non-proteolytic, pure form of urea, ammonia or nitrate are used as a source of nitrogen. When fermentation organisms are proteolytic, crude animal and plant raw materials like corn steep liquor, distillers dried solubles, casein hydrolysate, cereal grains, peptones, yeast extract, cottonseed meal, soybean meal, fish meal, linseed meal, peanut meal, etc. are used. So in nitrogen source, what we have discussed? Nitrogen source added to the fermentation medium may be organic or inorganic in nature. Organic nitrogen sources include proteins or amino acids. Inorganic nitrogen sources include inorganic salts like ammonia, ammonium salts, nitrates, etc. Further, some fermentation organisms are proteolytic and some are non-proteolytic. If the fermentation organism is proteolytic, that means that the organism has ability to hydrolyze complex proteins. So you can use complex protein sources as nitrogen providers. So if the organism is proteolytic in nature, you can add crude proteins, that may be plant derived proteins or animal derived proteins like corn steep liquor, distillers dried solubles, casein hydrolysate, cereal grains, peptones, yeast extract and different types of meals like cottonseed meal, soybean meal, fish meal, linseed meal, peanut meal, etc. But if the organism is non-proteolytic, that means it doesn't have ability to break down the protein, then you have to use simpler nitrogen sources such as urea, ammonia, nitrates, etc. as a source of nitrogen. Several crude nutrients supply carbon as well as nitrogen. For example, if you add molasses or corn steep liquor, then they will supply carbon source as well as nitrogen source. The nitrogen source sometimes influence the pattern of fermentation. This is also important. Suppose you want antibiotic synthesis. Then if you provide rapidly utilizable nitrogen source, it may inhibit the synthesis of antibiotics. Antibiotics are secondary metabolites. So they are produced during the stationary phase of growth of microorganisms. If you provide excess of carbon as well as rapidly utilizable nitrogen into the medium, the organisms will tend to continue in logarithmic phase of growth for a longer time, and therefore it will influence the pattern of fermentation and it will influence the antibiotic synthesis, because antibiotic synthesis will be inhibited. The ratio of carbon to nitrogen is also important. Fermentation media contain excess carbon. Excess nitrogen therefore induces rapid multiplication of organisms rather than product formation. Means if you provide excess of carbon and excess of nitrogen, the organism will tend to multiply and it will not accumulate the product. It will not produce the product. The next important component is inorganic nutrients. In many media, magnesium, potassium, phosphorus, calcium, sulfur and chlorides are required components. Media require many inorganic nutrients like chlorides, sulfur, calcium, potassium, phosphorus, magnesium, etc. Some components such as zinc, copper, manganese, cobalt, nickel, etc. are added in very small quantity. Crude media contain inorganic salts containing cations and anions along with the carbon source. Sometimes fermentation organisms have specific requirement of ions like magnesium ions, phosphates or sulfates. These requirements are fulfilled by addition of salts to supply these ions to balance the crude media. Sometimes during some fermentations we use organisms which have specific requirement. For example, they require magnesium ions, they require phosphates or sulfates. Then these are provided into the media in the form of inorganic salts. The concentration of single or multiple minerals is very critical in some fermentations. This may affect the production of secondary metabolites. Not only the type of inorganic nutrient, but the concentration of inorganic nutrient is also important in some fermentations. The next components in the fermentation media now we will discuss vitamins and growth factors. Crude media constituents provide enough amount of growth factors. So no extra addition of growth factor is required. If there is lack of any kind of vitamins or nutrients, growth factors can be added to the media by blending the natural constituents. Some amount of cane molasses is added to the beet molasses to supplement betaine deficiency. I will explain this. Many of the crude media contains the required growth factors. So although these growth factors are required for growth of microorganisms, they are present there in the medium as natural constituents of some crude components. But sometimes some of the crude components also have deficiency for one or more growth factors. For example, beet molasses, if you select as a raw material, then beet molasses is deficient in betaine. That means beet molasses does not contain betaine, and betaine is required for growth of organism. And what should be done then? You have to add pure betaine, or you can add some quantity of cane molasses into the beet molasses. Cane molasses contains betaine, so it will supplement the betaine requirement. The components added to supply the growth factors include yeast extract, beef extract, yeast autolysate, etc. These are growth stimulatory components because they contain growth factors. Which are these components? Yeast extract, beef extract, yeast autolysate, etc. Sometimes adding pure vitamin is more economical than adding a bulk of cheap source of vitamins. For example, if you want to add biotin, L-glutamic acid fermentation requires addition of biotin. If you want to add biotin, its quantity is critical for the product formation. You should not add excess of biotin. You should not add less of biotin. It should be added in the required exact component. It should be added in the required exact amount, and therefore it is usually added in pure form. Thank you.